molecular biology at the molecular level, that is, understanding how the molecules were organized, we can decide that it started in 1953. Some people say that the molecular biology is a combination of, call it the way it is, of crystallography and genetics. So that's where you have really the example, the first achievement. And what I want to show, because I will concentrate today not on the result and the structure themselves, but how to get them. That was the information that was used to give rise to this model. Of course, it was not. And these are the people involved.
Who's the lady?
Rosalind Franklin.
Ah, yes.
She collected that. Without that, these people would not exist. As a scientist, they would exist, of course, but
She wasn't her.
no. It's not a lack of respect, it's just put in the right perspective. And actually, that's the perspective what I'm telling you. You know, what is important in science is the result and also how to get the experimental evidences. So all my point of the discussion will be what kind of tools do we have now to get the molecular information. What does the crystallography do? Just very quickly. Crystallography is based, what you saw before, it was diffraction of not crystals, but elongated fibers. It's a fiber diffraction. But the principles are the same. The interpretation of the fiber diffraction was crystallography. Crystallography itself is with crystals. And don't forget that crystals contain up to 80 and sometimes more % of water. That is their concentrated solution structure. One tends to forget, you know, it's not solid state. It's frozen state. And don't forget, because what we are looking with other methods, like cryo-EM, is also frozen state. The only difference is that the crystals are ordered. And so you use that for diffraction. So one major step forward was in the 80s with the synchrotrons. At that time, you know, that was 30 years ago. It's not so long in terms of evolution. We're collecting data on these plates. And here is a diffraction pattern. You saw before, few spots. And that's one part of the diffraction pattern. That's from a virus. It's a large virus. On just that slide, you have several thousand diffraction spots. Altogether, we are dealing with millions of informations. At that time also, in order to build a model, we used this, what called Richard box. And the model was like that. That's a tRNA model, and the anticodon, that's the workstation we used. The crystals were grown. That's the next step. In the late 80s, first complex of protein and nucleic acid, RNA protein. These were the first real functional complexes. There again, another point that I want to make, the difference between a functional complex and a model complex. That's a functional one. Functional means you can run the reaction inside the crystal. That's the best proof that you have a functional one. Even if the kinetic is different, of course, it's much slower. You're talking in terms of hours or days and not seconds or tenths of a seconds. But it works. So what you see is really a functional complex. Here you have a complex. That's what the density looked like. That was the first map of this complex. You know this? That's a protein and that's a tRNA. Of course, it's low resolution, but the model, you see the crystals? That's where crystals, then you grow the crystals. That's history. Now we have more than, today I jump, 30 years later, more than 100,000 structures at the molecular level are available. More than 80% are from crystallography. But things are changing very quickly. And the one done by crystallography, they are available for you, PDB. Of course, that doesn't take into account all the structures which are not deposited. I mean, there are several thousand of structures done in the companies for drug design, development, things like that are not in that PDB. So let's move now from a step further. I'm going to compare, I mean, not to compare, actually, it's not fair, to bring to your attention what you can obtain by crystallography and what you can obtain now. Why is today another technique coming really important, becoming important study, that is cryo-EM. I will focus on these two. This is an example, 10 years ago already. It's not, it's a ultra high resolution that was developed for drug design. For drug design, you need very accurate models. This is among, I think, is the highest resolution. This number, you have the resolution, which means the limit of diffraction. And in turn, you will see that, so this number shows you how many diffraction data you have, and then the structure is here. Of course, when you lower here, it's called a map, density, electron density map, control at 8 sigma level, 8 electrons. So we clean out everything which is below 8 electrons range. And you see that all these spots here are experimental electron density. So at that level, you can see here the water molecule. You see all the heavy atoms, so-called heavy atoms. Oxygen is a heavy atom, carbon things, compared to hydrogen. But when you are interested in molecular chemistry, so analysis, this is from an enzyme, aldose reductase, which is rather important for medical applications. But I'm not going to talk about that. You want the most accurate structure in a meaningful conformational state. By meaningful, I mean you should have that one with different substrates and see what makes the difference. And if you know what these substrates make, for example, one would be an inhibitor and another one not, agonist, then you can compare what's different. And here you have, for the step, that's what we showed before. You see, here we do, by crystallography, one does difference map. That is, you make the difference between the observed map, the experimental map, and the map that you calculate with the model you have. That's a difference map. If your model reflects accurately the diffracted density, you should see nothing. If you see a peak, you have to interpret it, and this is what is missing, and then you look for what that means, you try to interpret. So that's the difference between the map I showed you before and this density map. Here you have the normal map, you see contour like before, and when you do an electron density map, you see everything is clean, but you have a spot here. And here is what you have with this electron density in green. I will not go into the details, but you see what is not the difference here. For example, here you don't see the heavy atom I was mentioning, but you see the rest. And at that level, with that accuracy, you see the protons. And then you can see the reaction and the things. I mean, you can interpret, sorry. That's, I would say, is about the limit that one can reach in terms of resolution with experimental data. Of course, that level of resolution cannot be obtained routinely. It was exceptional. There are maybe 5 enzymes, protein known at that level of resolution. But there are several thousand known at a higher level, or a worse level, sorry, we should say, 2, 3 angstroms, or 2 angstroms, which are quite good enough to interpret at the molecular level the mechanism. And until, let's say, a few years ago, 2 or 3 years ago, crystallography was the only experimental technique, method, that allowed you to reach that type of information. I keep in mind these crystals are solution frozen experiments. Solution. It's not, because very often people claim solid state. By solid state, well, it's like, no. is frozen liquid, and you can run the reaction. But in the last few years, and I will show you a few examples now, another technique emerged, which is a very old technique, cryo-EM, electron microscopy. Electron microscopy was used till 3 or 4 years ago to provide information of large complexes. That was its advantage. Could not be used for the small enzymes, but large molecules. But the resolution was limited to a medium average at best. That is 7, 8 angstroms, that was the limit, more often between 10 and 15. So you could get some informations, but not molecular level informations. This has now changed, and there are 3 main reasons why that new change, the revolutions in structural biology came around. One, and it's the major one, I would say, is the new detectors. When the same, the progression of crystallography, all the techniques, experimental techniques, these evolved with detection of the signal. That was true also for crystallography. You know, we use the first example I showed you were film data, and now they're detector, ultra-fast detector. The same philosophy. For EM, that was not the case. But a few years ago, a new generation of detector came, and this detector, what is different between these, mainly different between these detectors, before there were also films, and then other counter detector, that is detector detected the electron passing through, and you had to exchange the signal from the capture to another. You have to transfer. Each time you transfer, you lose information. Now these new detectors are counting directly. So you can go to the background, and you have a resolution, which is very important. So the resolution. Then now the 2 other revolutions that follow that, they do not as important, but they are becoming now as important, is the techniques to handle the samples that you see, called the realignment. We'll come back. And then the classification and reconstruction. And of course, now we can take advantage of all the software which has been developed and the approaches developed for crystallography. And so crystallographic methods are being implemented to refine and see. So it combines these methods. Here you have an example of this detector that translates quantum efficiency. You can have, I have on my slide, you know, I'm going, what I think, a bit fast compared to this, but you will have the references on my slide that you have, and you can go back and read the original papers and comment if you wish so. But here you have, I'm just spotting the main things. You have the differences here between the pictures that you can see. And if you go a perfect projection here, that's a ribosome. You can imagine, you will see from Marat's lecture next, I mean, it's a bit more clean than that, more complex. But if you look at an image at the beginning, that's how it looks like. And then you see here, you contact different contrast things. So the B thing is a simulation of the same image in cryo with a perfect detector. So if you would have a perfect detector, that's what you would see, starting from this. This is a model we construct. That's an image. That's not a real image. That would be a real image if you had a perfect detector, because you have noise, of course, and signal. That's a direct detection. And then you have you lose efficiency when you have different problems, the same simulated, but with all the receptor, the contrast is less, and that is much lower. So you lose contrast, and you lose things, and we'll see. Now we have this, I told you, the new generation things, and I will come back to this technical, we compare things, what that means. And the new generation of machines, and cryo-EM, this is the one we have in Strasbourg, Cryos, which was installed 2 years ago. Actually 2 years and 2 months now. It was in December 2013. That was first installed. And that is the type of images. So we had not the last, but the last but one generation of detector, Falcon detector. Now there is Falcon 2, and there is now an even newer generation, Falcon 3. Things are moving very fast now. And can we reach the atomic level? We give you an example that was reached. That was the first structure, the human ribosome. You will hear a lot more about ribosome. This is not to speak about ribosome, but with this machine, and you will see later, because that was an experimental set of data, 75,000 pixel. Here you see with the ribosome, it's easy compared to other to pick up the machine. So, because it's a very nice object for cryo-EM. And you have the classification starting from 2D. 2D you just select it from, and then you have a selection at the 3D level, which is much more refined because the molecule you can see it. The difference between these samples now, these are frozen crystals, frozen samples. So like the crystal, but a difference with the crystals, they are not ordered. In the crystals, they are part A order. Here, it just projected, but the idea, the assumption is, which is for most complex you will see real, not true, that's not true for everything, is the position when you project can be different. So although you always look at the same time, you can tilt a little bit, but then you can see that from different perspective. So you have different orientation. That's why you can have different classes. And of course, another major advantage of this approach, which I will come back, is when you project the solution, you have a mixture. We come back. Crystallization is selecting the proper sample. I told you it's an active and things, has a lot of advantage. Now with this one you have everything. So good and bad. The difficulty is there in sampling, it's not is to detect what is similar to each other. That's why you have the subgroup, subclasses. And you can see a complex in one given state and another complex within the same cell because you have an equilibrium where you have part of it missing or a different conformation. So a mixture, but within the same thing. You have taken your mixture and you look at all the components, provided you have enough things. And then, of course, one thing that you should look when you look at the papers. Now I will come back because today I will not only give you some information about the things, but to give you some perspective, to have also a critical analysis. And I will give you, that's why the title of my talk was Seeing is Believing. One tends to believe what is shown. So I'll tell you how to look, to open the door and go behind the door. But here this is shown, so it's not critical. You see, this color code showed you the resolution. That's cryo-EM. Equivalent in crystallography also. Not everything is at the same resolution because something which are disordered. So the resolution goes from 2.4 here at the core to 6 angstroms roughly for that sample, which is sold to high resolution. And the distribution of resolution is here with the major number is around 3.5. But you have a fraction of these below 3, and of course part which has. So you see that the inner core here, which is what you have more common feature and not different equilibrium, but because when you select, you can go up to an infinity of conformer. So you limit yourself at the level that you can handle, that is averaged. So when you average different conformation, when there are things different, you have an average. So you have lower information. This is, you see, other one. That's the density of the best part. That is something somewhere, I don't know exactly, around here. And you see that you can build at this level, the density is like a crystallographic high resolution density that you can interpret with a model. I must say at this point, it is important when you have large complexes. I will come back to that also later. It is important to have starting information. If you had to build from scratch, that is knowing nothing, would have taken a long time. When I show you the data collected in 2014, the paper published 15. That's the speed. And now I gave you the trend, and I will go more deeper into two main examples now that will take to show you the pro and problems, some pro and problems about the latest frontier on structural information. And I will finish with an example. Today, when we talk about structural biology, we are not talking only about crystallography. That was the field of crystallography, you see. From 1 angstrom, I showed to you, even better, to few angstrom resolution. That was the atomic resolution only. Cryo-EM used to be in that field, and now that's a bit optimistic, but okay, it is in that level. And you have other techniques which are evolving, light microscopy, I will not talk about, and solution study, NMR also, but NMR you focus on some specific things. You don't have the whole picture. Can argue about that, but that's it. And so you will see now structural biology is a combination, and structural biologists combine the source of information from different sources. That's why we talk not about X-ray crystallography, or we talk about integrative structural biology. And our goal now, we can really hope the new frontier is the molecules in the cell. We see functional molecules in the cell, so we are going to split. We'll come back to that. So we'll take two examples, one positive to illustrate, and one, you will see, halfway. So the positive one first is a long battle that the people in the field of RNA have known for more than 20 years now. Is the structure of the spliceosome. Crystal structure, not the crystal, the structure, the molecular structure of the spliceosome was a real challenge. I showed you before RNA, the first real molecular RNA was tRNA. There was a lot of structure about tRNA. We know a lot. They come back. It's still a key molecule, and there are still a lot of questions when you look at the function. Okay, I don't know, modification, let's see, and even I'm sure Marat will say a few things. There are a lot of things. But then when you moved away from these few examples, you had introns, we'll come back to that, and larger molecules. And for spliceosome was one of these originating. So how would introns, exons will be splits? And so it's very important for transcription, for transcription translation. And here you have an example, 3.8. So we'll give you 3 structures. It's amazing. More than 20 years work, close to 30 years, I would say, the beginning. You will see one of the founders of that really approach, Luhrmann, you see, that worked on the human one. He's part of that team. That was 30 years ago, I would say. I mean, roughly, it's the beginning. See. And in last January, 3 structures. The first one time, I don't know if it is the first, maybe in terms of submission, yes, but we are talking about a few months. Independent work. 3.8. You see now people are narrowing down to things. Of the ternary complex from the east spliceosome. Done, of course, by cryo-EM. That was really a very challenging attempt had been made to crystallize that. But at odds, and for reasons that we can discuss afterwards, no difference. Ribosome could be solved by crystallography. Crystallography was is one of the major results, everything. Spliceosome could not. You know, it's a term of different flexibility, mobility, so difficulty to assemble a functional molecule. But the molecules that are, the spliceosome is made of several proteins and RNA. Proteins, the independent modules, had been solved by crystallography. It's very important, because the interpretation of these maps would not be possible at the first level, would not have been possible without the crystallography structure. Here we come back to these integrative approaches. There is not one and then the other, forget about the other, not at all. And here you have a typical enough to be shown, we used to say, experimental data. If you look that very close, most of us will not see anything. But when you process in it, you got this classification. I mean, of course, there is a bit more than that, between that. And you see different projections, modules. And here is the plot of resolution. The 3.8 comes here. We say, now you have a drop, the real information content is there. The same representation. Here is the highest resolution between 3 and let's say 4.55 core, and then lower 6.58, and something difficult to appreciate, to interpret. And here you have that. 3.8, that's a reference. Science 2000, January, 2 months ago. It's not very old. You can go through and look. Now, I will come back to a paper which was published a few months before, 2015. Nature, 2015, by the group of Kyoshi Nagai. And work on the same origin, the Cerevisier East, it's an East spliceosome, again, this time at 5.9, 3.8, 5.9, 6 angstrom, let's put 6 angstrom. And here is from the abstract that you can go through, I have no time to go there. The structure provides crucial insight to the activation process. Essentially, it shows the architecture. At that resolution, it would have been a bit presumptuous to pretend that they have the molecular approach to see. So the architecture, that is putting together the module known by crystallography, is what they could extract safely. But when you have that, you can reach that model. So, you know, if you look at that level, we have no time to go in, but you go into the paper, you can look at the molecular level, but don't forget that each of these modules is a crystallographic structure that fits the density of the electron microscope. Okay, 50, that's okay. Now, yeah, this one is the last, of the, I don't remember, just a few months before January. It's 2015, the group of Kyoshi Nagai, and this is what real information was. And the imaging process that was provided, it is interesting. If you are interested, you should go. You don't get that in the main paper, you have to go in the supplementary material, but it provides the data, image processing. The same as what I showed you before. Data, a first screen for the 2D projection. You see it's not right, that much improved. Well, not fantastic, but you see between 6 and 4, it's a lot of data, you will see. And you start, don't forget, 367,000 particles that you select. You have an initial model to select, and then you process, and you have different conformers, so you classify. So you have different classes of structure. You don't know what is in. It's when you reconstruct, you know if they are all the same, but just viewed from the same angle, or different object. That is something which is different. You know, yeah.
So it looks to the human eye like these are not just different projections, but that the pieces can move.
That's another point. We'll come to that.
And so, is it having the crystallography on the parts that enables you to tell them?
Of course, we'll come to that.
No.
Of course, I was saying there may be different object that is complex with one more protein or another, or there can be different conformers. That is the dynamic. So when you froze that, you have one conformation and another. These are two objects. And at that time, so when you reconstruct, you see this, we'll come back to that. I think weaker, that's right. And in order to refine, just a little trick I give you, here you have the cryo-EM maps and the tilt pair validation. There are techniques to refine. And here you have gold samples. You see gold particles. Gold particles are like heavy markers. And you have the heavy markers without, and then you try to fit them there on the molecule. Here is the projection of the heavy marker. And so you try to refine your model so as they fit these give you the very accurate superposition of the orientation. And that's why you get high resolution compared to lower resolution. That's one of the techniques. I found it fascinating, actually, I should show. And you see here, unsharpened full map, the multi-body map, that's one of the refinement. You see a lot of more details. This one is growing up. So it's refined on the same object. I'm coming back to the other thing. Yeah, the resolution now, I told you, 2015 October, January or February 2016, the same group, the group of Yoshinagai, Nature, both were in Nature, now 3.7. When you go from 6 to 3.7, you can make 2 full Nature papers instead of 1. Crystallography, that is difficult. Now that was a joke. Well, after the joke.
Isn't resolution defined by resolution of these crystallographic structures that are...
No. It's a better resolution with EM data. I haven't checked. I think it's still the same detector. They were collecting data, and they were on their way to go, actually. They had no way of going even beyond that. But since the competition was publishing, they had to close.
So based on EM data, one can reach such a resolution?
Sorry? Oh, you start as soon as you can. There is no answer. You just keep improving, depending on the data you have, the quality of the data. It's a progressive thing. We'll see. I will come back to that. It's very important. It's really an interactive approach. But here you have the data. You remember 367 before. Now 475,000. It's slightly better. Same thing. Now if you would compare the two here, you see a better contrast. We are not talking about the same data reanalyzed. It's additional data. But here you see small complex, 3.7, more data. You remember before we had a lot less groups, so you can refine data, and you have the highest resolution, lowest, so the subgroup of highest resolution. Here you see you have now what I came back to your question, closed and open conformation. You see here? So we start now within this 500,000 reflection, a fraction of them is from a group in a given conformation. Another fraction is from a group in another conformation, each one with different things. And you can start. That's why they say here for this part, here the resolution is much lower because you have less particles which are common. Okay, so 4.3, 4.5. Again, now you're becoming familiar with that. The resolution, that's very high, the body, very high resolution. And then the further away you go, you lose resolution. And that's fix the limit. These are conventional factors to fix the limit of resolution that you can claim. Now the 3 orthogonal views of the same body. So that's the latest paper in Nature, the last month. And you see one view. I think the other one I have to speed up. At this point, you can start to look more deeply inside and look at interactions. But again, this time it's very useful to have the crystallographic data from subgroup to compare. Not only because you see, if the structure is ideally the same, it will fit perfectly. So you get the interpretation. But if there are changes, you see them. So they are double things. And then you can see complexly. So the interpretation used combination of at least two sources of information. But the revolution is that you can, with cryo-EM, you can reach that high resolution information. And now I have to speed up because of things. The third, I told you there were 3 papers. I should have said 4, because the East has 3 papers. One by the Chinese group in Science, 2 Nature that I just analyzed more deeply by the Cambridge group of Yoshinagai, and one at 6 angstroms, the other one 3.7. All 3 on East. Well, that's interesting, you can compare. I told you the most challenging one from the beginning was done by the work in Göttingen by Luhrmann group, Luhrmann and Holger Stark. Holger Stark is a cryo-EM. Reinhard Luhrmann is the molecular biologist who was the father of the work on this. So they came out with a 3D structure, the human. So it's the same thing from human. So it's very interesting now to combine the 3 because details you have. The resolution, 7 angstroms. So I told you before, you can see where you stand. So same ball game, but the lower level. I'm not saying this may not be the limit, but again, at this point, you can take advantage of the high resolution structure done by crystallography. And for example, so they have 2 references, the crystallography and the East structure, at least not the latest result of the East, but they didn't have, but the one at least of the last year, which was the architecture. And you can see that's highlighted by in the paper. For example, here is the real experimental density for the 3-way junction, the U4, U6. That's an RNA, as you see it. And that's the model realized density. So these kind of images are seen for the first time in DC. You have to realize that. 30 years of work to reach that. Of course, models could have been made, but that. And you see, all these are things where you have the crystallographic structure fitted in, because 7 angstroms you cannot. That's why 7 angstroms RNA structure looks like. You remember what I showed you before on the ribosome? At 3 angstroms, you could see the bases and the part. It's not part, but they are on their way. Why is that? It's because we have these flexible molecules. So we have hundreds of conformers in the solution, conformate. So we have to average. But if you have enough data, you go for 500,000 to 5 million, which will be possible. Then you improve, you select. But it takes time, you need computers and you need models. And here is the main solution that you gave. I invite you to go and read that in the paper. That appears in Science, well, last few weeks ago. How the spatial organization, they focus on some functional related things, and you can see how these are crystallographic models, put it in the right side. So you have the architecture, so you can interpret. And they can see, here you have the cryo-EM density. Here is the interpreted density. That's the East. So human, East, from the same perspective. You see there are differences. So now it becomes even more interesting when you compare things. You will see Mark will tell you more about the ribosome things when you compare bacteria and eukaryotes. But here human and East, they are both eukaryote, but very different. And so we are entering that region. Now, can I take 5 more 3 minutes? Because I told you we are on RNA things. I could have shown you an example, protein only, but I told you these RNA complexes are really challenging topics. And the protein-RNA interaction is part of the challenge, and the structure of the RNA itself. But here is another interesting example, the eukaryotic group 2 intron, lariat. The question is that, the mechanism. People are RNA field, they know the questions there, don't know the mechanism of excision there. So it's another side. I told you, I don't want to be negative, but I want you to come back with critical potential analysis. Here we are back. I told you how crystallography was essential and other things. Here is done by crystallography, not EM. So it's RNA only. So I skip the model on the lariat for time reason, but so that this is the question for the splicing. So here you have the insider, which is splitted, the exon here on the intron, and how this is the operation. So in order to interpret that, you need the structure of, ideally, all these three, but at least one of these. You see the interactions. Here you have the crystal structure, group 2 intron lariat, and the resolution 3.7, optimistic. So it's low for crystallography. I will come back to that later. Number reflection, 31,000. You have to compare with those, 31,000 from the same conformer. You know, it's not, it's 10 times less, but you have a unique conformation. Don't forget, in terms of information, you have all the redundancy. That's okay. But here you have lots of magnesium ion. When you see that, actually, if you read the paper, it's another Nature paper. It's not because in Nature that everything is fine. But it's another of my take-home message. So Nature 2014 is not very far. High resolution. Could show you maps of RNA structures at high resolution where you see the magnesium, the tRNA. Looks very nice. But here, I was wondering, had a problem. The resolution is rather poor, so how could they fit that? This is technical, rather poor, but okay. And what's more, the B factors. B factor is called the temperature factor in the refinement. Before I told you on cryo-EM, when you have different conformers and you average out, the more flexible they are, the less you see, they loosen. In crystallography, where you have that problem, also means you fix that by the B factor. The higher the B factor, the more flexible the part. And so the less density you have, or the less meaningful the information is. That's where I got a bit rise. For the RNA is 200. For the ligand ion, magnesium, 200. The water, 200. When I was doing my postdoc with Michael Rossman a long time ago, this number would never have gone further. I would have been thrown out. So here you have a density. That's what is published, density modified map. What is the real density? Calculate, because now you have to provide the data. So if you are interested, even if you're not knowledgeable things, you ask a colleague or a friend, structural biology, go and pick up and calculate the map, and can I see it? And that's the most interesting part, but you don't see any magnesium, things like that. That's a critical part, the exon. Here is the exon, how it is built. Every time you go, you do the same experiment you did, and you will look sharply for your density. There is none for most of it. So again, when you have an information content, even if the result is important, you have to look at the critical part. What is the question? What is the experimental answer? If it is not provided, usually it's provided in most good paper. If there is a problem, you go back because the data have to be provided. And when you see things like that, you better look carefully. Now, I wanted to, but I hope, I just wanted to teach you the things. It's not because it is in a paper, even in a nice, high impact factor paper, that this is true. We'll have the solution very soon because there is another structure available. I have not seen the density. but I know that it is the real one. And that's why I could convey the message without with information and important to you. Come back to now from molecule to cell I did before. Today we use integrative structural biology, where we combine all that. And I didn't speak of all these methods, which are solution methods, transient scattering, solid state NMR, ultra high resolution photonics. That is entering really a challenge to look in the cell, where are the complexes? And there you isolate the complexes and you look at them with other techniques. Tomography. Tomography is jumping also by cryo-EM. And then I'm finished, Glauco.